And now we want to talk about this transfer RNA, its design, and what you're seeing with regard to the design. You have three depictions of the same thing, of the transfer RNA, and each of them are good to look at. It's the third one, the simplified one, that will show up on all the subsequent images that you see in this teaching time and also in the book. But what you have is you have a string of nucleotide bases. And that's in the two-dimensional one. You have a string of nucleotide bases that have been folded in a particular shape. It looks kind of like the nave of a church, to tell you the truth, every time I look at it. But anyway, down at the bottom of it, in the two-dimensional one, you have this anticodon area, and that's the three actual nucleotide bases that then are going to correspond to the messenger RNA. So all the rest of it is the portions of the transfer RNA that are important in terms of its structure. It has kind of an L-shaped structure. That structure is going to fit into an enzyme, which will show up on the next slide. So that structure is important. It isn't just throwaway information, but it's these three bases at the anticodon, which will actually match up with a messenger RNA. And then at the superior aspect of it, you have a binding site for an amino acid. So you have this this amino acid attachment site, and it's so designed because you actually have a place where that amino acid can bond temporarily to this transfer RNA. The transfer RNA carries one amino acid. Transfer RNA also has then one anticodon at each ends. What happens is transfer RNA consists of a single strand of RNA with only about 80 nucleotides, whereas messenger RNA can have hundreds of nucleotides. There is a little bit of give with regard to this anticodon. We'll talk about that in a slide coming up. So this is the enzyme structure that's responsible for attaching that specific amino acid to that specific transfer RNA. And there's 20 of these. So these are called, let's see, where's the word? It's an amino acyclic transfer RNA. You see that synthase. You see the term there. It's quite a mouthful. But what it is, it's a complex of about 20 different enzymes. As a matter of fact, 20. Each one is unique to that amino acid, and it's also unique to that particular transfer RNA. So only one transfer RNA will fit into this enzyme, and only one amino acid will fit into this enzyme. And what will happen is the transfer RNA will go to this enzyme as well as the amino acid. The amino acid will be attached to it, and then with the help of energy, ATP, and then the base will be released. We call this the aminoacyclic transfer RNA at that time. So the amino cycle, we'll call it the amino cycle. It tends to be less of a mouthful there. Will be released to then be able to bind to the messenger RNA. There is a little bit of a wobble here. So this term wobble is something that is used scientifically because that third base of the codon allows for more than one transfer RNA to bind there. So you may in one particular sequence, you may have an A at the third end, and but you have a transfer RNA that has a G that's matching up with it. So at that third end, there is a little bit of shift with regard to these transfer RNAs. They found this out because in bacteria makes multiple proteins. There are 64 different combinations with regard to messenger RNA RNA codes, but only about 45 of these transfer RNA. So they know that this is what's going on, that there's a little bit of give with regard to that third unit in the codon. And then a little bit more about ribosomes, because they are what's going to facilitate this whole process. Remember, there are two units, large and small. Now, they are not joined. They become joined and they become a complex. But what happens with regard to some of our antibiotics work by actually targeting the bacterial ribosomes. So we know the bacterial ribosomes are slightly different than eukaryotic ribosomes, so they can actually pick those out as opposed to the human body cells. Going on to more about this ribosome, you have three sites. You have an A site, a P site, and an E site. The A site is the initial binding site. So called because it's named after that aminoacyl transfer RNA binding site. And then you have the P site, which is the site where the polypeptide is being created. We know that's the case because you have this exit tunnel. And when this transfer RNA comes in, it's going to have that amino acid hanging off the top, which the amino acid needs to hang off the top so that it can actually bond to another amino acid and form this polypeptide chain. And then you have the exit site. And each of these is part of this process of initiation, elongation, and termination, so that eventually the messenger RNA is going to be in the middle of these two pieces of ribosomal units, and the transfer RNA is going to, if the messenger RNA was made upstream, the transfer RNA is going to be made downstream. So initiation brings together messenger RNA and transfer, and so we're looking at the left picture there, and you just have the small ribosomal subunit. You have that binding to the messenger RNA. You have the initiator transfer RNA coming in. We know it's the initiator. It's the codes that match up with the start codon on the messenger RNA. And so you have this binding together, and then you have ATP coming in, or GTP, very similar to ATP, but GTP coming in and attaching the large unit to the small. So basically creating this this vice, and all of this together becomes the translation and initiation complex. So the sum total of the small unit with the messenger RNA on it, the large unit attached with a transfer RNA in there, and the start amino acid attached. During elongation, amino acids are added one by one. On the growing end, we call it the C-terminus end. The initiation starts at the N-terminus end, and grows along the C-terminus end. Each additional protein adds to the elongation requires three things, three steps and factors for each of these. You have to recognize the codon. The peptide has to bond, and then translocation, and then translocation proceeds along the messenger RNA. So let's go through this in terms of image. And by the way, there's some great graphics that you have them on your Mastering A&P. You have some great visuals, and much of this stuff might be easier to understand if you actually see it in a short 1 or 2 minute clip. But what you have is you have your initial terminus, your initial site, and then another transfer RNA is going to come in with an amino acid. That then, with the help of GTP, is going to bond that amino acid to the polypeptide chain that's there, that then is going to, that first one, the one that's in the A site, is going to stay. The one that's in the P site is going to move out to the E site and then out. So what happens is they come in the A site, move to the P site, then to E site, and then out. And as they move along, this polypeptide chain, the amino acid that was brought to this complex, is bonded to the polypeptide chain, and the chain moves along. Termination's a little bit different with regard to the transfer RNA. What happens is, remember, we get to the stop codon, and what you have is you have a transfer RNA that brings a water molecule. So a water molecule is brought instead of an amino acid. This water molecule then releases the polypeptide because of the chemical interaction that takes place, releases the polypeptide, and then the whole assembly comes apart. So in the termination is because a water molecule is brought instead of an amino acid. That water molecule then hydrolyzes, you might say. Every time water comes in, it hydrolyzes, and causes the entire complex to then come apart. Freeze the polypeptide, an entire complex comes apart. And then you have kind of a summary of all of it here. Remember, we started with transcription. RNA is transcribed from DNA. That happens in the nucleus. It's then modified, it's processed, it's spliced, and you also are going to put ends on it. So you're going to put a 5 cap and a poly A cap. It then is going to leave the nuclear envelope, and it's going to meet up with the small unit of the ribosomal RNA. The ribosomal RNA, with the help of a start transfer RNA, so you have the start transfer RNA coming in, it's going to meet up with the small and then the large and the transfer RNA unit. That's going to make all one structure. The messenger RNA is going to move through the ribosome, creating this polypeptide. That's the translation part. Then finally a water molecule is going to be brought in. The whole thing's going to come apart. The polypeptide is then going to be released. The polypeptide chain is going to be released into the cytosol, and that might be finished. That might be the end of it. Except for it's not the end of a protein, but understand, remember, you have free ribosomes, and then you have ribosomes that are attached to endoplasmic reticulum. Most of the free ribosomes make proteins that are used by the cytosol itself. It's the endoplasmic reticulum that tends to make proteins that are going to be packaged to be exported outside of the cell. So in exocytosis via vesicles. So this polypeptide, basically after it's formed, it can start turning into it can start doing the folding with regard to making a protein, the folding, and then the three-dimensional structure, and you can also have several polypeptide chains getting together to form a protein, but that can happen all together in the cytosol, or it can move on to the endoplasmic reticulum. Now, the number of ribosomes that can translate a single messenger RNA simultaneously is, you have a special name for this. It's a polyribosome or a polysome. It's just basically a ribosome that's enabled to make a cell to make copies of a polypeptide very quickly, because you have multiple polypeptides being made at the same time. And so, because when you think about it, some of these proteins are very necessary immediately. You might eat, so you might eat cheese, and then you need a protein made to actually digest it. We'll pick this up on the next one.